Hi, everybody. We are INBRAIN. We are a health tech company using graphene to decode brain signals into breakthrough medical solutions. We are 50 people with more than 35% of women. We are a spin-off of the Graphene Flagship, which is a consortia that the European Union put with 1 billion euros to actually bring graphene to market. We raised significantly funds as to get to first in human this year, and now we are looking for a Series B round of 55 million. We also managed to get a very big collaboration with Merck on bioelectronics, so combining the neuroelectronics potential with the bioelectronics potential, we can get access to a 25 billion business opportunity. Now, this is what legacy technology of platinum and iridium have been able to do, decoding the size of a zebrafish that, as you know, is tiny, and it's only 100,000 neurons. Now, the brain has nearly 100 billion neurons, and we need to understand it because 1 out of 3 people actually have a neural-related disorder, and 30% are refractory to medical treatment, building a big cost for our society. Every leap in humanity has been linked to a key material. From a Stone Age to Silicon Age today, we believe that bidimensional materials like graphene are the next breakthrough for neurotechnology. Now, graphene is a Nobel Prize winner material. It's the thinnest material known to man at an atom thick, yet it's 200 times stronger than steel, a very good conductor and very biocompatible. And as the Nobel Prize winner said, game, it's about the physics that it's able to deliver. Legacy therapies based on platinum and iridium are just big, limiting the adoption of neurotechnology. They have also very poor resolution that need very precise implantation of very small nuclei of the brain, and therefore limits the outcomes that we can get on these patients. And also, usually, they don't relate to the environment data of the patient, and therefore don't drive personalization. This is exactly what we can change. Graphene drives miniaturization to a cell-like size. We can drive also very high resolution in a bidirectional way, so actually increasing the outcomes, increasing the adoption, but also bringing new biomarkers of the environment of the patient and connecting with modern electronics to drive full personalization. We've been decoding the brain nuclei by nuclei. Like that, we will never make it. But neuro-related disorders are relating to a circuit on the brain that is pathological. This is just an example of the first indication. We are doing Parkinson, epilepsy, and then a speech, more on the brain-computer interface side. But you can see how it would work. We are placing a cortical brain-computer interface module on the motor cortex in this example, and then a subcortical module deeper in the brain. And we are looking at the synchronization of this network, in this case, the nigrostriatal pathway for Parkinson, detecting pathological biomarkers and correcting them to increase the percentage of time in therapy of a particular patient, therefore increasing the outcomes and reducing the side effects. Same for other disease areas. This is exactly how our platform reflects the therapy. We have a cortical brain-computer interface module that can have up to 1,024 contacts, just like Elon Musk, and a subcortical module that has also quite high resolution, all connected in a powerful intelligent decoding unit that also connect to the external biomarkers of the environment of the patient, driving that full personalization. I won't go into inervia, which is the one that we developed with Merck, but the vision is to connect the central and the peripheral nervous system, because at the end we have one nervous system. You see, this is how it compares to legacy technology. Legacy technologies on platinum and iridium has millimetric sizing, reducing the potential on the outcomes detection. And also, if you compare to the graphene technology, we can go submillimetric with a lot higher resolution, 10,000 times smaller than the standard of care. So what we do is 2 main things. Because we can inject 200 times more charge injection at a very low impedance, we can reduce battery management and actually reduce the systems, so driving the miniaturization and the higher adoption. But also, we can drive 10 times better signal resolution and therefore outcomes. These are just 2 examples on recording, so reading, and stimulating writing. So on recording, you see that actually we can read bands on the brain that metals cannot read. Therefore, it's like having a microscope that suddenly sees that bacteria, so we can create that antibiotic. We are seeing things that the others cannot see, and therefore deliver therapies that the others can't. So that's on recording. And stimulation we have compared to other polymers that are also coming into the market, like the one from Neuralink, and we see that we have very much a very superior stability. So with stimulation, the material doesn't degrade like others. Now, the first application has high resolution, but the second technology that we are applying on the cortical brain-computer interface goes to ultra high resolution. And there we can read every single frequency of the brain from 0.1 hertz to kilohertz, where actually on the 0.1 hertz, on the low frequency bands, we have bands that actually are the ones that precede an epileptic seizure. So by looking at these bands with metals cannot see, we can actually anticipate on that epileptic seizure and stop it, because stopping a seizure once has started is very difficult, but looking at minutes before that it starts, it's very easy to stop. Now, on the graph below, you see the recording of the 1,024 contacts live in an animal, and this has been published in Nature. We have proven also the pathological state control in a Parkinsonian model in rats, and now we are doing also in large animals. And we can read single unit activity, LFPs, so we can also modulate the classical beta, but we believe, and I'm sorry I cannot show that data, that the closed loop and the treatment of these diseases is not only in single bands, but a combination of bands that actually relate to that circuit pathology. And all that requires a lot of signal processing, and that's why we are also coupling and developing a data hub with clinical applications and research applications. With Series A, we have achieved a lot. We have actually driven an FDA pre-sub that has been proved to be very positive towards a commercial application in the US with the cortical brain mapping acute cortical interface. And we have also driven the subcortical to large animal trials. So the only two milestones that are pending to do is the efficacy studies on large animals of the subcortical interface and the first in human in cortical. And just recently, we also received from the European Commission 17.5 million that will be part of the Series B, making that 55 million possible and actually having more than 50% of that. We have a very experienced team coming from powerhouses like Philips, Medtronic, Onward, but also material science. And then on the clinical side, we have also a very global clinical board. And recently, we have also incorporated, and we will announce soon, a vision board with a Nobel Prize graphene winner actually joining us, and other important people like David Eagleman, which is a very distinguished Stanford neuroscience scientist. So we build credibility, but also visibility. We just won last month the best innovation company in 2022 in Spain. We are looking for these partners for Series B, and as I said, we have more than 50%. If you feel this is of any interest, feel free to actually reach me after this presentation. Thank you, everybody.